guys, welcome to the final lecture in beta oxidation, fatty acid metabolism, and talking a little bit about electron transport chain today, and getting into some more of the biochemical structures, processes, and everything associated with what we've been talking about. So when we left off last, we were talking a bit about muscle carnitine and how muscle carnitine is activated, what it does, how it functions, and now we're going to talk a little bit about how muscle carnitine kind of acts as a sensor for fuel selection. I know we kind of talked about that a little bit on the last lecture, but keep in mind that activation of muscle carnitine would indicate that there is a high level of fatty acids being taken into the cell to be metabolized, and a reduction of carnitine activation and activity would basically mean that there is a low amount of fatty acids being taken into the cell, which would then essentially tell us that we are metabolizing sugar, or we are in an anaerobic anaerobic capacity. And exercise intensity is going to play a big role in whether or not we activate carnitine and what carnitine is doing. So just a couple more facts on carnitine. It's important to understand that there's about 100 times more carnitine than there is CoA concentrations, okay? And that's because CoA will fix itself to a precursor like acetyl or acyl, where carnitine will transport it and then drop it off, okay? So we have more of the carnitine than CoA, so that we can assure ourselves that the transport of these necessary fuel molecules get where they're supposed to go. And at a moderate intensity of work, between 50 to 70%, we know that both glucose and fatty acids are activated at that. So if we're looking at 50 to 70% of VO2 max, obviously at 50% of VO2 max, we're going to have a lower activation of glucose metabolism. But then when we increase the intensity, going from 50 to 70% of our VO2 max, we're going to increase that sugar utilization, thereby down-regulating carnitine. So higher intensity, lower carnitine activation, lower intensity, higher carnitine activation. Okay, so they're inversely related. So, and what I mean by that is, if we have high activity, high exercise, then we have low carnitine mobilization, where if we have low exercise, we have high carnitine mobilization. So that would be what we call an inverse relationship. And then when we increase activity above 75% of our VO2 max, obviously fatty acid utilization is going to decline, and we're going to kind of shift back into those anaerobic systems and start relying on glucose and glycogen once again. Okay, so I basically am trying to draw you a picture of what all these variables are doing during different phases of exercise. So if you want to look at how carnitine is, how it adapts to exercise, we got to think about this shuttle as being something that will upregulate itself when we get in shape, and then as we get older, it will downregulate itself. Also, when we are kind of moving and shifting into disease states, carnitine is one of those proteins that can be targeted for lower activity and downregulation. So if we begin to exercise, and we exercise regularly, carnitine activity and expression can be upregulated, which means then we have more of this transport capacity, right? And as I mentioned before, a lot of people will take exogenous carnitine to try to increase more expression of carnitine, but it really depends on the stimulus of exercise. So we know if the body is producing it naturally as a response to continuous exercise, well, that's going to be more effective than just consuming carnitine and hoping that you're going to increase fatty acid oxidation. That's not necessarily true. So in training, we will have several adaptations. I'm going to kind of repeat myself a couple times here. When we begin to exercise more, we'll get more expression of this CD26 fatty acid transporter. and that will basically multiply. So we'll have more expression of these transporters that will embed themselves into the membrane, so that the DNA will basically produce more of it within the sarcolemma, and fatty acids will then be able to enter the cell at a much faster rate. We will also have more CPT1 expression, so we'll basically upregulate more CPT1, which will help convert acyl-CoA to fatty acid acylcarnitine. So we get more of these. We get more carnitine. Carnitine becomes activated more quickly, and carnitine will also do its job much more efficiently with exercise. And the same thing is true with disease states. So in an adaptation, a healthy adaptation that's based upon exercise and exercise stimulus, we'll have more of these things that will express themselves in the skeletal muscle, which will help you metabolize fatty acids more effectively. However, just like we talked about in many different proteins in this course, if we have a disease condition that is beginning to develop, then especially metabolic diseases, these things will decrease their expression. So we'll have less of these transporters, right? We'll have less of these fatty acid transporters. We'll have less carnitine. We'll have slower carnitine. We'll have less CPT1. We'll have less CP2. And obviously what that's going to do is completely slow down this supply chain here, which brings fatty acids into the mitochondria. So ultimately, we won't have the mechanisms to break down fatty acids and get them into these derivatives that we need in order to create energy. So what can be problematic there? Well, what could be problematic there is when we increase fatty acid consumption through a Western diet and we don't exercise. Well, this conveyor belt system here that you see, it begins to back up. And when it begins to back up, yeah, we can take fatty acid into the cell, but we'll get all these unprocessed derivatives here that will just start floating around in the cell because we don't have the correct machine or the volume of these machines to help convert this to this, and this to this, and this to this. So this supply chain begins to back up, and instead of breaking down fatty acids and getting it into the TCA cycle and then into the electron transport chain to create ATP, we have these derivatives that just don't get processed and they float around in the cell, and they can cause problems within the cell. So we can get excessive acetylcarotene, we can get excessive acyl-CoA, we can get excessive fatty acid acylcarotene. So these things become unprocessed, and then they become problematic. All right, so if we exercise, we upregulate. If we don't exercise, we downregulate, and then we start to run into metabolic perturbations that cause more problems in that cell. So here's a little bit about some research that they've done on taking supplemental carnitine, exogenous carnitine, and I just kind of put this up here to show you that studies have been conducted on this. Let me see if I can zoom in a little bit more here. Studies have been conducted on this since the 80s, and really not much has happened after the 90s. It's something that we don't really research anymore, is exogenous supplements. But you can see that most of the research here, not really compelling evidence. You can see that this group, they found a slight but significant increase in VO2. Here they saw very little benefit in exercise performance. This group here, in 40 athletes, they saw an increase in VO2 max. Well, maybe in elite athletes that excessive amount of carnitine would play an important role in shuttling fatty acids. Here we saw no increase, here we saw no increase, here we see increase. Lipid utilization in muscles during exercise. So there's really no conclusive evidence or really any solid evidence that says that this works 100% in everyone that takes exogenous carnitine. And what a lot of people do is we'll run to GNC and say, hey, I want to lose 15, 20 pounds. I'm just going to take this carnitine. I'm not going to exercise. I'm going to eat what I want. And of course, that's not going to work. If you're an elite athlete, sure, those additional milligrams of, or these actually, they use grams, so they use a pretty heavy dose. That's not milligrams. A super physiological dose, which is 3 or 4 grams, that's a lot of carnitine, might give you a slight advantage. Okay, but again, when we exercise, we activate those transcription factors. Those transcription factors to communicate with the DNA. The DNA tells the human body to produce more of a protein, or in this case, several proteins that will aid in exercise performance. And once you stop exercising, those transcription factors will turn off. The stimulus will not turn them on anymore, and your body will degrade those extra proteins, because if you have them and you don't use them, then they can become problematic and lead to metabolic complications as well. So there is a crosstalk, as I mentioned in the first slide. There is a crosstalk between fatty acids and glucose. And this crosstalk means that carnitine is going to be the mediator that determines what fuel is being used and what fuel is not being used. So, for an example, in disease conditions, patients that have carnitine deficiencies also experience metabolic stress. So that tells you that carnitine plays an important role in metabolic switching, right, or metabolic flexibility. So the capacity and the ability to convert energy via glucose or via fatty acids. So carnitine does play a role in that. We know that an intracellular accumulation of acyl-CoA derivatives have been implicated in the development of insulin resistance. So let's go back to this slide. If we have an excessive amount of acyl-CoA, okay, or acetyl-CoA, it doesn't matter, either one of them, an excessive amount that cannot be processed, and maybe it can't be processed from here to here, or maybe it can't be processed from here to here. If carnitine is not doing its job, that leads to insulin resistance. Now, why is that important, and what does that have to do with crosstalk? Well, because we're talking about fatty acid oxidation here. We're talking about fatty acids being used as a primary fuel, and if carnitine dysfunctions and these derivatives can't be converted from this to this or from this to this, because carnitine uses both of them, and I would advise all of you to pay a lot of attention to this slide, because this is something I'm definitely going to ask you guys on an exam. If carnitine fails at making those conversions, that causes insulin resistance. What does that have to do with glucose metabolism and crosstalk? Well, what it does is it impacts glucose uptake, because obviously if you have insulin resistance, then glucose can't get into the cell to provide energy in using glucose, right? So that is how carnitine, in that particular incidence, crosstalks between fatty acids and glucose, okay? So there is evidence there that shows that there's crosstalk between those two metabolic systems. In cell culture, right? So when we're doing cell cultures and we're controlling the environment, one thing that's interesting about cell cultures, and this is why cell culture is not a really strong means of proving a hypothesis, because cell cultures, they have very limited contractile machinery. So they do have some myosin and actin, but it's not like what we see in a human muscular tissue, right? And cell cultures, you can't contract them unless you add caffeine to the culture or you stimulate them with electricity. So you can artificially contract them. But when you don't contract cell culture, there is a low abundance of carnitine, right? So that tells you that muscle contraction plays a role in carnitine expression and activity, right? So if we look back here, let's go back. We were talking about how exercise and adaptations to exercise increases fatty acid utilization. Well, I told you that it's going to upregulate more of these transporters. It's going to upregulate carnitine, increase carnitine activity, as well as increasing carnitine expression, which means we'll make more of it. Exercise has a positive impact on CPT1 activity, CPT2 activity. So we know all of these things are increased with exercise. Now, the inverse to that would be these cell cultures that we were talking about. If they're not contracting, that is basically representing a sedentary lifestyle. So if we looked at a cell culture model and there's no contraction and the cells aren't doing anything to basically use energy, Well, that means that carnitine is then going to be downregulated. And if it's downregulated, that means that fatty acid oxidation is also going to be downregulated, which means that the cell culture is going to shift to an primary glycolytic metabolism. So it's going to try to use glucose more than anything. And one of the reasons being so is because carnitine expression might be downregulated. Okay. So, when we have a decrease in these enzymes, their function, their capacity, their expression, we are going to have a decrease in fatty acid oxidation and an increase in glucose metabolism. That's fine and dandy if we're exercising and we're remaining flexible, we're remaining or keeping that metabolic flexibility. That's fine and dandy, but when we are sedentary, we do not want to be consuming glucose at rest. That leads to, as I said up here, right, metabolic stress. We do not want to be in a position where we're sedentary and we're experiencing metabolic distress. All right, so we spent a lot of time on bringing fat into the cell, talked a lot about how exercise increases the capacity for fatty acid metabolism. We talked about a couple of key proteins that makes that happen. We know that with exercise stimulus, we upregulate those proteins, but with sedentary lifestyle, we decrease them. So if we don't use it, we lose it, essentially. So now let's talk about the beta oxidation process. Now, keep in mind, beta oxidation happens before the TCA cycle. So in order for us to move this potential energy into the TCA cycle to ultimately produce a lot of that NADH and FADH2, which is going to then be the major currency to produce energy in the electron transport chain, these energy derivatives must undergo further transformation. So we have another layer of regulation here, okay? And again, appreciate how the cell regulates the conversion of energy so that the next organelle or the next series of biochemical processes in the mitochondria can receive that new currency and transfer that currency into more potential energy. So we spent a lot of time here. We looked at all this. We looked at all these major players. Now we're going to look at beta oxidation. And the major thing that I want you to get from beta oxidation is that we are now converting a acyl-CoA to acetyl-CoA. And acetyl-CoA is what is going to be allowed to introduce the TCA cycle. So acyl-CoA cannot enter the TCA cycle to produce those electron carriers, NADH and FADH2. So in the mitochondria, these fatty acids, or these derivatives of fatty acids now, are going to undergo beta oxidation. And beta oxidation is basically going to be removing carbons in the form of acetyl-CoA, and it's going to basically transfer that into potential energy in NADH and FADH2, okay? So we're going to go from acyl-CoA through beta oxidation to acetyl-CoA, and then acetyl-CoA can then enter the electron transport chain. So let's look at this in a bit more detail. So this is a very simple representation. I want you to keep in mind that we have four carbons, okay? We're looking at acyl-CoA here. And I'm going to draw your attention down to here and notice that once a 4-carbon acyl-CoA undergoes this transformation, we're going to get an acetyl-CoA, and pay attention here. Now we have a 2-carbon acyl-CoA, which is going to go back through this process again, and those carbons are going to be removed. So this is the third step in beta oxidation or fatty acid oxidation. And what's going to happen here is this is a catabolic process that is going to remove hydrogen ions with electrons while producing acetyl-CoA, all right? And we can see that those electrons are going to be tagged onto these electron carriers, right? So beta oxidation, just like the TCA cycle, is going to produce these electron carriers that are going to go to the electron transport chain, right? So that's why fat is so robust in producing energy, because as it goes through these different layers of metabolism, we're producing potential energy in the form of FAD and NADH, FADH2. Sorry, apologies about that. So this says it's removing hydrogen ions, but keep in mind, an electron always follows a hydrogen. And we can see here that it's going to remove 2 carbons. And in this process, we are going to create an FADH2 and we're going to create an NADH. All right, so that's where those electrons are going. So this series of reactions where we take an acyl-CoA and we run it through beta oxidation produces acetyl-CoA, which can enter the citric acid cycle, and it also produces this potential energy as electron carriers. Okay, keep in mind that the citric acid cycle cannot accept acyl-CoA. It has to accept acetyl-CoA. So again, when you're exercising, we have the ability to increase the activity of some of these enzymes. And this guy right here is an important enzyme. A lot of research has been done on this. They call this HAD. And this enzyme will increase its capacity to process fatty acids coming in. It could also be downregulated. And what's great about this is it increases its capacity. It also increases NADH production, right? You stop exercising, this guy will slow down, which means this will slow down, right? So the stimulus will basically dictate the metabolism and the efficiency of the metabolism. And this NADH, as well as this FADH2 will go into the electron transport chain to produce energy. And this byproduct here, which is acetyl-CoA, will enter the electron transport chain. Okay, and we know that ultimately this and this will produce ATP, and we know that this will produce more of these in the citric acid cycle, which will also produce ATP. Okay, so let's look at this in a bit more detail. So here is a kind of a zoomed in process. Now don't get caught up with all the nomenclature here with the chemistry. Let's just look kind of at the steps, okay? So we have 1, 2, 3, 4. We have 4 kind of major steps in here, okay? Now you can see FAD, it does not have the extra hydrogen or the electron added to it. So when fatty acyl-CoA enters beta oxidation, the first thing that happens is a hydrogen ion is removed, an electron is going to follow that, and it's going to be tagged onto this FAD. FAD will turn to FADH2, and that will go to the electron transport chain. Now, what is interesting about this is this first process right here directly produces energy by being sent to coenzyme Q in the electron transport chain. If you don't know what coenzyme Q is, we are going to talk about it in a little bit. But that is the enzyme that is going to carry potential energy from the protein complex 1 to protein complex 2. So we also call coenzyme Q an intermediate. So that's why this picture is here, because this is basically saying that in this first step, when acyl-CoA comes in and it interacts with acyl-CoA dehydrogenase, this enzyme here is going to remove an electron and a hydrogen, and it's going to tag it on to FADH2. FADH2 is then going to communicate with coenzyme Q and produce ATP. All right, so that's why this is kind of an important, picture. Okay, once this happens, acyl-CoA dehydrogenase is going to create an intermediate which is called trans-2-anoic-CoA. This is not important. Do not worry about this, okay? This is not important. Don't worry about this. This is not important. Don't worry about this. This is important. So let's kind of look at this, okay? So basically, acyl-CoA comes in, it undergoes and interacts with an enzyme. It gets turned into a derivative, just like glycolysis. There's another enzyme waiting here. This enzyme takes this in, turns it into a derivative, and then there's another enzyme here which basically takes this derivative and turns it into something else. But when this enzyme interacts with this derivative, it does something similar that we saw up here with acyl-CoA dehydrogenase. So it is going to then remove this enzyme. This is hydroxylacyl-CoA dehydrogenase. That is I call it HAD, H-A-D. This enzyme is going to receive this derivative, and it's going to remove you can see it here, this hydrogen ion. It's going to remove it, and it's going to tag it to NAD, and NADH. It's going to create NADH. This NADH is going to undergo oxidative phosphorylation in the electron transport chain and produce ATP. Now, when we exercise, we are going to increase HAD protein expression and activity. So this is one of the enzymes that is super sensitive to aerobic exercise. And if we increase this enzyme activity, we accelerate the movement of these derivatives through beta oxidation. So these things move much quicker through it. And that makes sense, right? Because not only are we going to increase fat uptake while we're exercising, but we got to make sure that beta oxidation and the citric acid cycle can accelerate these molecules faster through their process so that we can create ATP at a much quicker rate, okay? So this guy here is very susceptible to both exercise and to sedentary lifestyle, all right? And just like this guy, FADH2 is going to interact with coenzyme Q to create ATP, the NADH here is going to interact with the electron transport chain coenzyme complex 1, which is going to create ATP. And if you don't know where complex 1 is, I will show you that momentarily, but it is the first transmembrane complex in the mitochondria that accepts NADH, okay? So if you don't know what it is, you need to kind of pick up your game a little bit and make sure that you understand where that protein is. Now, after HAD receives this derivative, it's going to create one more derivative. It's going to interact with this last enzyme here, and now we are going to produce the necessary acetyl-CoA, which is going to enter the enter the citric acid cycle. Also, note that we started with 1, 2, 3, 4 carbons. I told you that through the beta oxidation process, we cleave 2 carbons every time a molecule goes through the beta oxidation process. Now, this has been really kind to us, and it's only given us a short chain fatty acids, which only contains 4 carbons. But when we're dealing with long chain fatty acids or very long chain fatty acids that have 20 to 22 carbons on it, those are very robust and rich in their ability to produce energy. So imagine if this had 24 carbons, 22 carbons, and it cleaved 2 carbons every time it went through. Well, that means every time the new version of the original fatty acid went through beta oxidation, we would be creating these energy molecules here, FADH2 and NADH, right? So in this example, they were very kind and they just showed us a 4-carbon molecule. So you can see that once 2 carbons have been removed, here's 1 carbon, 2 carbon. This is acetyl-CoA. Acetyl-CoA is going to go into the citric acid cycle. Well, there's 2 carbons remaining. So now we have a 2-carbon acyl-CoA, and this has to be processed to create acetyl-CoA. So this molecule will then return through beta oxidation and undergo this same process and produce more FADH2, produce more NADH, and produce 1 more acetyl-CoA, okay? And that then this molecule, once converted to acetyl-CoA, will then enter the electron, I keep saying the electron transport chain, sorry, enter the citric acid cycle, okay? So this is beta oxidation. So what I urge you to keep in mind here is that fatty acid, acyl-CoA, will enter the beta oxidation process. It is super, super dependent on how many carbons it has. Once this molecule goes through beta oxidation, it will cleave 2 carbon molecules to create an acetyl-CoA. It will create FADH2 as well as NADH, very similar to glycolysis. And then the remaining carbons will have to go through that process again. So if we have 20 carbons, if we have a long chain fatty acid coming through, the first time that fatty acid goes through beta oxidation, there's going to be a molecule here that now has 18 carbons, and it will have to go through again. And then it will come through and have 16 carbons, it'll go through again, come through, have 14 carbons, go through again, and this process will keep repeating. So what I want you to pay attention here is this step, this enzyme and what it produces, Know that this enzyme produces a byproduct that will directly help in energy production by interacting with coenzyme Q. I will show you that relationship in a little bit. This guy's not important. This guy's not important. This guy is important, okay? Hydroxyacyl-CoA dehydrogenase. That is an important enzyme. Know that this enzyme is susceptible to adaptations in exercise. It's also susceptible to sedentary behavior, but also know that this guy here, this third enzyme, will produce electron carriers, NADH, which will interact with complex 1 in the electron transport chain, thereby producing ATP. And the other thing I think is really important for you to understand is that the byproduct of beta oxidation is the conversion from acyl-CoA to acetyl-CoA. Acetyl-CoA will enter the citric acid cycle, and then the remaining carbon acyl-CoA will go through the beta oxidation process again. So that is all fair game on an exam. Be ready to be familiar with this slide and be familiar with it as it is. I'm not going to try to trick you in any capacity, but I might ask you, what if this happens? How, if A, B, and C were to happen, what would that do to this beta oxidation process? All right, so kind of be ready for that. Now, just to kind of connect the dots a little bit for you guys on the beta oxidation process and those complexes that I was referring to. This is the mitochondria. You can see that the mitochondria has several transmembrane proteins. This is complex 1, complex 3, complex 4. We have some intermediates in between these complexes, which are coenzymes, which will carry those electrons from protein complex 1 to protein complex 3. Protein complex 1 in the inner membrane of the mitochondria is what is going to receive that NADH and going to take that electron and that hydrogen and produce energy. So you can see here that the NADH that was produced in beta oxidation is now going to interact with this protein complex 1. All right, so let's go back. I told you that this NADH that is basically created at this HAD enzyme here, it is now going to enter the well, it's already in the mitochondria, but it's now going to interact with the matrix and this protein complex 1 right here. So I told you guys there's a complex 1, complex 3, complex 4. I also told you that FADH, which is going to be produced at this first step in beta oxidation, where we can see this acyl-CoA dehydrogenase enzyme. This enzyme is going to create FADH2. And to return back to this slide, you guys, this is kind of a poor representation because you really can't see the detail here, but just keep in mind that this FADH2 is going to interact with a complex that is right here. So the big takeaway here is that when NADH and FADH2 are created via beta oxidation, those electron carriers are going to move into the mitochondria and begin to start producing ATP. And then the acetyl-CoA is going to enter the citric acid cycle and begin to create more of these electron carriers, which is going to create more ATP overall. So that's why even a short chain fatty acid is going to have a much more robust capacity of creating ATP than a single molecule of glucose. So, and again, the further we get into medium chain fatty acids, long chain fatty acids, and very long chain fatty acids, once they enter the beta oxidation cycle, every time they enter and re-enter, they will have 2 carbons removed and energy and electron carriers will be generated to create energy. So that is beta oxidation. Just keep in mind where that step is within the mitochondria. Understand that fatty acids have to be brought into the muscle cell via this transporter. We have to have these conversions of these derivatives here in order to pass the outer and inner mitochondria membrane and get through the inner mitochondria membrane space via CACT. This derivative here must be converted back to acyl-CoA, and that's done through CPT1. And of course, carnitine is involved in that process. And then once acyl-CoA is in the matrix of the mitochondria, it has to enter beta oxidation. So essentially, in order to get this guy into the mitochondria, it has to be converted, and several enzymes have to help transport that across the mitochondria membranes, and then it can be undergo oxidation. So let's talk a bit more now about mitochondria, and we'll kind of look at this electron transport chain with a little more detail, and we'll get into a bit more nitty-gritty in the upcoming slides. So just in case you're not familiar with the mitochondria, let's just go over some basics. It is involved in calcium ion control. It is not just a powerhouse of the cell, but it is also a it is involved in cell signaling. It also undergoes and plays a role in apoptosis. So it has several functions other than just creating energy. As we get older, we have a decrease in mitochondria population. And as muscle begins to undergo atrophy, we also have a decrease in mitochondria density. Mitochondria function gets worse with age. So this is something that is definitely very sensitive to lifestyle, very sensitive to age, chronological age. It's very important in communicating with other organelles, and it's also very important with crosstalk between glucose utilization. So keep these things in mind. So if I were to ask you a question about how the mitochondria may or may not be involved in crosstalk with glucose, these next few slides might be important for you to pay attention on. So let's just look at the layout or the blueprints of mitochondria. Let's look at some of these components of it, just in case you're not familiar with it or you need a refresher. So we have ribosomes. Ribosomes, of course, since mitochondria creates its own DNA, well then mRNA, which is not messenger RNA, but mitochondria RNA, will get converted into more mitochondria protein within this ribosome. There's some of these don't matter. I'm not going to ask you anything about granules. Understand that we have the inner membrane space, right? So we have the outer mitochondria membrane, the inner mitochondria membrane. We have that inner space here. As I mentioned, if we have ribosomes, well, obviously the mitochondria has its own DNA, and you can see that here. So it has its own DNA, which will produce more mitochondria. AT synthase particles, this has to do with enzymes that we are not really going to talk about much at all. It has a matrix. With the matrix here is the central component, right? This innermost component here. Outer membrane space, or I'm sorry, outer mitochondrial membrane, we just talked about that. And cristae, this is essentially, you can see this folded region here, right? So the mitochondria has this component, which is almost like a scaffolding protein, right? It creates more surface area within the mitochondria, very similar to the small intestines. And, the electron transport chain is embedded within this area here as well. And this plays a major role in energy production. So most importantly, when it comes to the cristae, just understand that it increases the surface area of the mitochondria, where electron chains can exist and help create energy. So that's where the electron transport chains rest. So let's talk a little bit about the citric acid cycle. So we left the beta oxidation cycle, and now we're coming into the citric acid cycle, and this essentially does the very same thing that we saw in beta oxidation. We are passing electrons. So we're going to move, in this case, we have acetyl-CoA coming in, and we are going to start removing electrons. We're going to oxidize acetyl-CoA from these acetyl groups here, and then we're going to create more of these electron carriers. And this is something that you should have seen several times by now. We know that once acetyl-CoA comes in, we produce a lot of these electron carriers here. If this were a normal class where we were taking exams in the classroom, I would absolutely expect you to know every single one of these derivatives here, such as citrate, isocitrate, alpha-ketoglutarate, succinate, fumarate, malate, oxaloacetate. You would be expected to know all of these and to draw them, but that serves no purpose in this type of class, because you can certainly look that up online and score very well on an exam. So I'm not going to ask that of you, but I might ask you a critical thinking question here about how this works. So generally, when we have this production of energy here, these electron carriers, just like the electron carriers that we produced through the beta oxidation cycles, they're going to basically interact with the electron transport chain, and they're going to interact with specific proteins within the electron transport chain. So this one here is going to interact with the first protein complex, and the FADH2 is going to interact with the second protein complex, and we'll talk about that in a little more detail as well. What is important to understand is this one here. When I talk about crosstalk, you can see that mitochondria function is sensitive to glucose and carbohydrates. So in this bullet point here, it says the maximum rate of oxidative phosphorylation is markedly reduced if available carbohydrate stores are depleted. Now, why is that? So I want you to take a moment and think about why the absence of carbohydrates would reduce oxidative capacity. And the reason being is because if pyruvate is converted into acetyl-CoA, and pyruvate, which is produced through glycolysis, enters the mitochondria, then it will be converted to acetyl-CoA, and it will undergo oxidation, and it will help create these electron carriers here. So if we are depleted of carbohydrates, then we are depleting the mitochondria and the citric acid cycle of half of its available potential energy, because now we don't have carbohydrates and fatty acids contributing to acetyl-CoA. So again, keep in mind, when it comes to metabolism, we're not talking about a single fuel being on all the time. We are talking about ratios of fuels being on all the time. If you remove carbohydrates from your diet, if you go on a ketogenic diet, you are doing a disservice to mitochondria function and oxidation and energy production. So that's why a lot of people that are on ketogenic diets, they just kind of feel weak, or they don't feel like they have much energy, okay? So let's look at this in a little more detail. When we have a change in mitochondria enzyme function as a result of exercise, so we have exercise-induced adaptations, when it comes to specifically the contributions of these enzymes in energy production and increases in VO2, they really have a modest increase in VO2. So what I'm saying here is, if we're exercising and we're gaining those wonderful exercise-induced adaptations, these enzymes within the mitochondria are sensitive to that process. So oxaloacetate is something that increases its function. It increases its ability to recycle and add 2 carbons onto acetyl-CoA. Citrate is known to have increased function and concentration, and succinate also is known to have increased function and concentration. So when we exercise, we can accelerate the conversion of some of these products as a result of constant training, right? So basically, the citric acid cycle will function more efficiently and faster and create more robust concentrations of these electron carriers when we exercise. And the same thing is true when we stop exercising. This whole process begins to slow down. And when we become sedentary, well, then that means that we're not using the muscle, and these things slow down even more, right? So again, if we don't use it, we lose it. So let me go back to the previous slide. So when we look at the function, the increased enzyme function within mitochondria as a result of exercise, it really only plays a modest role in energy production. And basically what most studies show, if you took the mean average, it only really has like a 20% increase in VO2 max. And what a lot of research has found is that, yes, it's wonderful to increase mitochondria enzyme function and capacity, but that is still limited by the ability to deliver oxygen to the muscles. So even though we increase the enzyme function and we increase the efficiency of the citric acid cycle, that impact on VO2 is very small, because the enzymes are still limited by oxygen delivery. So oxygen delivery is a greater limiting factor than enzyme function, okay? So keep that in mind. But one of the benefits that do come from increased mitochondria function, increased citric acid cycle efficiency, is there's smaller disturbances in homeostasis. So that means when you start to exercise, your heart rate doesn't increase as much. Your temperature doesn't increase as much. You might stay in a specific energy pathway for a longer period of time. Exhaustion will take longer to reach. So when we talk about exercise adaptations and increasing the efficiency of the citric acid cycle, the most robust effect that this process has, or increasing the efficiency of this process has, is it keeps you in homeostasis longer, right? So your body doesn't have such a massive response to exercise, right? So if you were in shape and you were running next to your friend and your friend wasn't in shape, and of course when we say in shape, we're referring to adaptations to exercise. If they were struggling and huffing and puffing and sweating and you weren't, well, part of the reason you're not is because as a result of exercise, you have increased the efficiency of this process. And then most importantly, again, we're talking about crosstalk. I've been talking a lot about crosstalk in this relationship between glucose and fatty acid metabolism, is that when we have higher enzyme levels in the mitochondria, higher enzyme function, more efficient enzyme function, well, that helps us get into fat oxidation faster. It helps us do it at a higher rate, and thus we spare muscle glycogen, and we also maintain euglycemia, right? So we spare this stuff and we maintain euglycemia because we don't dump all of the glucose from the blood into the muscle to be utilized as energy. So that's wonderful. So if you're an endurance athlete and you've been exercising, and as a result of exercising, you've increased your efficiency here, you've increased your efficiency in your beta oxidation. And if I were to go back to, let's say, let me find this slide here. If I were to go back to here, and as a result of exercising, you're also increasing transportation of fatty acids into the muscle, you increase CPT1 expression, CPT2 expression, well, now you have heightened your efficiency in fatty acid utilization and ATP production. So you can see how all these players work together. So let me go back to this. And if you increase the function and the expression of all those proteins and all those enzymes, well, then you get into that aerobic capacity much faster. And if you get into that aerobic capacity much faster, you utilize less glycogen, you utilize less blood glucose, and there's less of an oxygen debt that needs to be paid back at the end of exercise.